Welcome back to this video. Today in this video, we are going to continue on the topic cell division, and we're going to go to the third part, which is on meiosis. So this is our part 3 for chapter 6, cell division. And the outline of this video is that we're going to discuss about what is meiosis and what are the issues of cell division. So it includes mitosis and also meiosis on our human health. So what is meiosis? The definition of meiosis is a process of cell division that occurs in reproductive organs, okay? In reproductive organs. So in males will be testes, and in females, the reproductive organ will be ovary. And then it's a process that going to produce gametes, okay? So the reproductive organs produce gametes that contains half the number of chromosomes, which is the haploid number of the parent cell. So the parent cell is having a diploid number, which is 2n equals to 46, okay, for a parent cell. But the gametes is going to have half the number of the diploid number. So the haploid number is n equals to 23. So that's the difference between mitosis and also meiosis. The meiosis, you can see here, this is the overall view for meiosis, where they're also having a interphase, and then they're going to undergo a meiosis 1 and meiosis 2. Last time in the last video, when we mentioned on the mitosis only consists of 4 phases, right? But now in meiosis 1, in meiosis, we have meiosis 1 and 2. So total here, we are going to have 8 phases, okay? And the outcomes will be a daughter nuclei, all right? A daughter nuclei, which is haploid in number. So the need for meiosis, why we need to have meiosis? Okay, so the aims of the meiosis is actually to form gametes through the process of gametogenesis. So it's for reproductive organs to produce gametes. And mitosis is actually for the somatic cells, which is the rest of our body cells, to produce a normal cell, okay? But this one, meiosis, is just for our reproductive organs to produce gametes. And this meiosis is to ensure that the diploid chromosomes number of organism, when two haploid numbers, one from your father and one from your mother, combines together, and then only becomes a diploid chromosomes number. Okay, but just remember that the outcomes of our meiosis, the daughter nuclei, is always haploid in numbers, which is n equals to 23. And then the very important aims for meiosis is to produce genetic variation. This is why me and you are different from the perspective of genetics. Okay, so first, as I said just now, we have meiosis 1 and 2. So all the processes are the same. We have prophase, PM80, just same like our mitosis, prophase, metaphase, anaphase, and also telophase, but going to separate into 2 for meiosis, which is we have 1 and 2. So first we start out with our prophase 1. So in prophase 1, the things is going to be the same. The chromatin shortens and it thickens, and the chromatin will become visible chromosome, which is visible under our microscope. And then this is an important keywords here for prophase 1. The pairing of the homologous chromosome. Homologous chromosome means that the chromosomes that are having the same size, same length, okay? So this is a pair of homologous chromosomes. You see the blue color and also the red colors. And the pairing of this, they're going to undergo a process. This is known as a synapsis, okay? Synapsis. So you see, they pair together, and this is known as synapsis, and we form a bivalent. So the process of synapsis will form these whole things here. We know it as a bivalent. And then crossing over is another important keywords for prophase 1, which is to ensure the exchange of genetic material to produce a new combination of genes. So we can see here in our pair of the homologous chromosomes here, there are, you see the little space that we circle here. So this place is actually where the process of crossing over happens, okay? So they kind of exchange genetic material to produce a new combination of genes, okay? And the areas of crossing over is a verb, is the name for the process. And the area for the crossing over is known as chiasmata, right? So that's the two important. So there are 3 important keywords that you need to remember for prophase 1. The first one is synapsis, which is the pairing of homologous chromosome. And number 2 will be crossing over, which is the process of exchanging genetic materials. And number 3 is chiasmata, which is the area for crossing over. So that's all, okay, for prophase 1. And moving on to metaphase 1, remember that during the last video for mitosis, I tell you that the important keywords for metaphase is line up or align. So here in metaphase 1, the homologous chromosomes, after exchanging the materials, they're going to arrange at the equatorial plane, which is produced by the spindle fiber. You can see we have our centrioles here at the poles, just seem like the normal cell division, and they form spindle fibers, and all these chromosomes are going to align at the equatorial plane. And then the sister chromatids are still tied together because the centromere has not separated yet. So in our meiosis 1, so in our metaphase 1s or the next processes, the sister chromatids are still together. They will not separate from the centromeres, okay? So this is another thing you need to remember. So in metaphase 1, they arrange at the equatorial plane, and then the sister chromatids still tied together, okay? Because the centromere is not yet separated. So next, we're going to talk about Anaphase 1. So Anaphase 1, the spindle fibers, which is this yellow lines here, they're going to contract and shorten, and then they're going to bring the homologous chromosome to the poles, okay? Going to pull them to the opposite pole. And important keywords here, the chromosomes is still made up of a pair of sister chromatids, and is different from our mitosis, where they are separated at centromeres and the sister chromatids are pulled apart from the at the centromere. But in this case, the chromosome is still made up of a pair of sister chromatids, so they are still combined at the centromere. They didn't separate, okay? So that's another important features for meiosis 1. And next, moving on for telophase 1. So in telophase 1, which is the last phase for our meiosis 1, the chromosomes arrive at the opposite pole already. And then now the polar cell, so this considered as one polar cell, and polar cell contains a number of haploid chromosomes, which is n equals to 23. And then the spindle fibers will disappear, and the nuclei will reappear, and the nuclear membrane will reform again. And this telophase 1, or the whole meiosis 1, is succeeded by cytokinesis. Do you still remember what is a cytokinesis? A cytokinesis is a process of the divisions of the cytoplasm, okay? And then it will produce two daughter cells after meiosis 1. So, which is haploid in number. Always remember that the outcome, the product for meiosis is haploid in number. And the outcome for mitosis, which is what we talked about in the last video, is diploid in number. And then the DNA does not replicate in meiosis 1. So that's why we are having a haploid number of a chromosome rather than a diploid number. Okay, so moving on after cytokinesis, after producing two daughter nuclei in a haploid number, then we're going to proceed with prophase 2 here, okay? So far, so clear. And then in prophase 2, I'm going to tell you that in meiosis 2, everything is going to be quite similar as mitosis, okay? Which they're still having prophase, metaphase, anaphase, and telophase, the PM80. And all the processes is quite similar to mitosis, okay? Just that the meiosis 1 is different. And meiosis 2 is almost same as our mitosis. And then prophase 2, the nuclei and also nuclear membrane, they're going to disappear, and the spindle fiber going to form by the centrioles to pull the chromosomes to the pole. And now the chromosome is made up of sister chromatids, same. And you can see that in this, like for example, we colored this chromosome as green color, but you see you see here there's a little piece of red color here. This is the products of the crossing over. So that's a little piece of a products from the crossing over, which to ensure that there's genetic variation for every gametes, okay? And then spindle fibers start to form. That's all for prophase 2. And then next, moving on to metaphase 2, which important keywords here are line up or align. So chromosomes are arranged at random on the equatorial plane. And then metaphase 2 going to end when the centromere is going to separate. See, in this process is same as the mitosis. The sister chromatids going to separate at the centromere. But for meiosis 1, the metaphase 1, the centromeres does not separate. So the sister chromatid does not separate, okay? So in metaphase 2, the centromere is going to separate, and then the sister chromatid is going to be pulled, going to be separated and pulled to the opposite pole. And next, moving on the Anaphase, where the centromere is going to start to separate, you see. And now we no longer call this as sister chromatids. Sister chromatids look like this. And this, the sister chromatids pair separates and they move towards the opposite pole. So this is no longer call it as sister chromatid, we call it as a chromosome at this stage. Okay, so in Anaphase, they're going to separate at the centromeres and they're going to be pulled to the pole. And lastly, for Telophase II, which the chromosome arrive at the pole already, and then the number of the chromosome for each daughter cell is half the number of the parent chromosome. So go back to our important features for meiosis, which is all daughter cell or all daughter nuclei is going to have half the number of the parent chromosome, which is always haploid in number for meiosis. Okay, and this meiosis II is going to be continued by cytokinesis and produce 4 daughter cells. Okay, so there's a little bit difference that I want to talk about here, which is in mitosis. After cytokinesis, we produce 2 daughter cells. Okay, they are diploid in number. But for meiosis, after cytokinesis, we're going to produce 4 daughter cells, which are haploid in number. This is the very important difference for mitosis and meiosis. Okay, so you can see outcome here, we have 4 haploid daughter cells or 4 haploid daughter nuclei. All right, that's all for meiosis. It's a little bit complicated. Maybe you can rewatch that until you understand. And in the last of this video, we're going to talk a little bit on the issues of the cell division for human health. So we know that normally, usually, in our human cells, the cell division is going to be normal. There will be some regulations to make sure that our cell division is always in the systematic manner. But sometimes uncontrolled cell division is going to happen. And this is going to lead to formation of tumors, which is going to lead to cancer later. So cancer is actually due to uncontrolled cell division. And our cell division, our cell didn't divide in a systemic manner. It's going to divide in an uncontrolled manner. So this is going to lead to tumor, okay? So that's how cancer comes generally. And any abnormality during the divisions of meiosis, okay, going to cause a genetic disease such as Down syndrome. I'm sure lots of you heard of Down syndrome. So another terms for Down syndrome is trisomy 21. Trisomy 21. So what is trisomy 21? So you can see here, this is the karyotypes or the whole set of the chromosome for human, which we have 23 pairs of chromosome. So you can see 22 are our somatic cell or our body cell, and the 23rd is our sex chromosome, which consists of X and Y. So this is a male. And then trisomy 21 or Down syndrome is where at the chromosome set of 21, there is 3 chromosomes. That's why the word tri, T-R-I, comes from means 3. Okay, so trisomy 21 is where we have 3. We have 3 chromosomes, and in this chromosome set 21, we have 3 chromosomes, and that's going to cause problems such as Down syndrome. And the baby, we have problems such as mental retardation and having some problem with his IQ as well. So that's why a proper or a controlled cell division or meiosis is very important for our human health. Okay, if otherwise it's going to cause cancer or even genetic disease like Down syndrome. There are still a lot more, but for your level, just know that Down syndrome and also cancer. Okay, so for summary of this class, we're going to recap a little bit. So we actually talk about what is meiosis, which is the outcome of meiosis is always 4 daughter nuclei that have a haploid number. And then we also talk a little bit on the issues of cell division on human health, which is cancer and also some genetic disease such as Down syndrome. And last, I have some question for you here, which how do you compare and contrast mitosis and meiosis? I hope from my last video, mitosis, and also in this video, meiosis, you are able to answer this compare and contrast question, okay? So that's all for today's video. Thank you very much, and I will see you in the next video. Bye-bye.